Next up, we're going to hear from Randall Kaye, who's going to update us on their product, which notoriously I'm going to mispronounce medication name, Bexicaserin, which was previously known as LP352. So Randall, thank you so much.
Thank you. It's nice to see everyone. I wasn't so sure how to pronounce it either because I'm originally from Boston and I didn't know whether you put the R in there, so I didn't know whether it was like Bexicaserin or Caserin, so I'm probably still going to use LP352 because it's just easier. With that in mind, I think the earlier part of the discussions today were very much focused on approved medications that have gone through a journey and have been very successful. As we're getting into the afternoon sessions, a little bit more about this sort of new wave. I've entitled my talk as we're leading into this new wave of hope. But there's a challenge in drug development. You heard it from our colleagues at Takeda. Sometimes medications don't work as well as we hope they will, but we learn a tremendous amount along the way. Every time we're in the midst of developing or discovering new medications, we take tidbits, we take information, we learn what people did well, and we learn when there are relative failures, and we use this information to make our studies a little bit better and a little bit better. So I'm going to give you one of those examples and tell you a little bit about our compound. It actually is pronounced Bexicaserin, so just in case you really wanted to know. So Bexicaserin is the next generation 5-HT2C receptor agonist, which does sound like a little bit of a mouthful, which has greater selectivity and specificity. So what does that even mean? We've designed a medication in mind that focuses on a receptor that we think provides the key efficacy. We have a slightly different approach from some of the other precedent compounds. Is we wanted to focus on the receptor that we know contributes to efficacy, and if you know your compound is interacting with other receptors and you don't know what they do, or you do know what they do and that receptor is affiliated with an adverse event, our idea is why don't we design a molecule from scratch that only interacts with one specific receptor and then find out whether that might be a better approach. And so we're in the kind of early part of this sort of journey, and I'll share with you where we are and what data we're starting to see. We know that the medication that we have in development, it penetrates the brain in a dose-dependent fashion. That's important because you want to make sure that the medication gets to where it needs to be, especially when we're talking about central nervous system disorders. We also know that modulation of this receptor can potentially suppress this hyperexcitability that often can lead into seizures. And it's based on an initial company and work that's been done over the last 20 years on what's referred to as GPCRs and optimization of this 5-HT receptor pathway. The molecule you see on the bottom portion of the screen, it's a new molecular entity. So we didn't, in the sort of experimental medicinal chemists that we have, we didn't just make some minor tweaks. We kept experimenting with really novel molecules. So this doesn't even look anything like some of the other precedent compounds that are well known to interact with this serotonergic system. But that's kind of like the hypothetical stuff, right? So we have this compound, now we need to understand what potential impact it can have from both a safety and an efficacy standpoint. So I'm going to take you through a phase 1b/2a study. It's called the Pacific study that we released top-line results in the earlier part of this year, and then talk to you about some results that we just released last week that we refer to as an open label extension, a little bit longer period of time that we were investigating and following patients that were treated with Bexicaserin. So very crowded slide, but just to focus you a little bit on the upper left-hand corner. This is a study in participants with a broad variety of developmental and epileptic encephalopathies. So yes, it did include patients with Dravet, but it also included patients with Lennox-Gastaut and we allowed for patients with a variety of other underlying DEs, up to, I think there are about 25 named ones at this point, but there's probably a lot more. So it's a slightly different approach that we've seen in the study of individual DEs. And we'll get into more of why we did this approach a little bit later. So the study design allowed for a variety of patients to come in. They needed to have at a minimum of 4 countable motor seizures per month in the preceding 3 months. And then during the screening period also needed to, again, as documented in a seizure diary, have a minimum of 4 countable motor seizures. Patients were randomized in a 4-to-1 ratio, so that's 4 patients treated with Bexicaserin versus 1 on placebo. It was done in a blinded fashion. And we did a dose titration based on tolerabilities. It started off with 6 milligrams. This is a liquid formulation that's administered 3 times a day. If they tolerated, they went up to the next dose, up to 9. If they tolerated, they went up to 12. They could go up and down for the initial period, and then would enter into the maintenance period where they were on study drug for a total of 60 days. They were given an option of potentially rolling into an open-label extension study. If they chose not to, they were entered into a dose titration. I'm giving you a few extra details because I want those who are kind of thinking a little bit maybe of considering having their loved ones involved in clinical research. The steps that I just described is very typical for what happens for patients that are enrolling in studies. It is a commitment. But I hope you'll see as we talk through some of the data and information, it's an important commitment so that we can develop and move forward with discovering new potential medications. I described overall this being a double-blind, placebo-controlled study. The focus in these early-stage studies is on safety and tolerability. We do blood evaluations to look at how the body is handling it from a pharmacokinetic standpoint. And then we're also able to look at efficacy as an important outcome because of the seizure diaries that were completed. This study was run in the US and Australia at over 30 sites. And we did enroll patients down to the age of 12. So we had an adolescent population, which comprised about 20% of the population. The remainder were adults. So I'm going to start with just giving an overall what we saw from a safety standpoint. Overall, 52 patients were enrolled in the study, 43 on Bexicaserin, 9 on placebo. So the safety data that I'm going to describe to you is we refer to as the safety set. So that's anyone that had any exposure to medication whatsoever. And then a full analysis set looks at patients that went into the titration period, also into maintenance. And those are the ones that we looked at efficacy because they had a sufficient amount of time enrolled in the study. So what do we see from a safety standpoint? There were 3 serious adverse events that were deemed not related to study drug, but these occurred because all these participants ended up being hospitalized. 2 episodes of a fractured ankle, unfortunately in the same exact patient, 2 different episodes or events. Also increased seizures were noted in 1 patient and hospitalization due to constipation. Probably more helpful information relates to the adverse event profile and discontinuations. During the titration period, 16% of patients treated discontinued during that up-titration period, that first 15 days. But you'll note only 4%. So this is just a small number of patients discontinued during maintenance. So it's important as we learn more about this medication, what's the best way of titrating, of getting patients on this medication so they can successfully go through the maintenance period. And then the most frequent adverse event that was observed was somnolence, and that was the most frequent reason for discontinuation. On the right-hand side of the slide, you'll see the most common adverse events that are listed, and these are pretty typical with anti-seizure medications, especially those that interact with the serotonergic system. So a little bit about efficacy. So we look at the experience that patients have in the study over the full treatment period. So that's that up titration, those first 15 days, plus the 60 days during maintenance. So it's a relatively short period of time. So we have to focus more on things that we think would be impacted with a drug therapy in a short period of time. So we focus predominantly on these countable motor seizures. We observed a 59% drop in or seizure reduction over that time period versus 17% on placebo, with a delta or a mean difference of 42%. These are important results from a preliminary standpoint and gives us the knowledge that to encourage us to go further into clinical development. I'll just make one quick note. We do intend to initiate a global phase 3 study by the end of this year based on both the safety and efficacy results. But I'm going to give you a little bit more insight because I know a number of you have loved ones with Dravet and you may be sort of curious of these subpopulations. So on the left-hand side of the screen, these are very, very small numbers, caveat, but the 3 patients that did have Dravet overall had a 74.6% reduction. In that treatment block is the way we refer to them. None of those patients were randomized to placebo, so I don't have any placebo comparative numbers for you. And then we looked at both LGS and the D group as a whole to get a sense of what those comparisons were. You'll note about a 50% reduction and 65% in LGS versus DE, and placebo responses in that 17 to 32% range. What's probably more important to notice here is the mean difference, that delta, is almost the same in LGS and DE. And that's important because as drug developers, we start to think that maybe there may be a different way of studying developmental and epileptic encephalopathies. Maybe we can start to research these a little bit more broadly rather than these single studies that just focus on an individual indication. And so on the next slide, I'm just going to show you, it's what I refer to as a dot plot, and it just shows the individual responses. And you can see on the left-hand side all the way over the responses in those patients with Dravet. 2 out of 3 had very impressive responses to drug therapy, and then a lot of similarities when you compare the LGS and DE other group. So that's the data that we were sitting on as we were making our plans to move forward into our pivotal studies. But we had some additional results that we talked a little bit about and released last week that I wanted to share with you. So this is the open label extension study. There were 41 patients that completed the double-blind portion of the study. And all of those patients decided to go into the open label extension study. And so I have interim analysis of 6 months. So this gives a lot more insight to what happens from a safety standpoint when people have prolonged exposure to a medication for a longer period of time, but it also allows us to see what happens from an efficacy standpoint. And I'll just give you kind of a broad summary of this. I mentioned that all of the completers and participants at the end of the study opted to go into the open label extension study. The breakdowns were 3 in LGS, 3 in Dravet, 20 in LGS, and 18 in the D other group. And what we saw, and you can see this on the right-hand side, is a sustained response over that 6-month treatment period. So in the big blue bar, we're seeing very similar responses relative to what we observed in the Pacific study initially, and a very favorable safety and tolerability profile. I'll just make a note that's rather interesting. That middle bar in placebo, there were 9 patients that were randomized to placebo. This is their treatment response during the treatment period. I mean, you'll see it's very, very comparable to the initial Pacific study for those who were originally randomized to Bexicaserin. And a few important points about that placebo group. They all successfully titrated up. They entered into the maintenance period, no discontinuations, and they were successfully able to stay into the maintenance period. And we're seeing just a very similar treatment response. So we're on track to initiate this global phase 3 program later this year. I'm going to skip ahead to this one quick slide. This allows us to look at the treatment over each individual month. So the last data I just showed you was what do we see for that full 6-month time period. This gives us a little bit more of a view of what happens after the first month of treatment, the second, the third, and so forth. And you see a very quick relative response in the placebo group that came very, very close graphically to what we'll see in those that were originally randomized to Bexicaserin, and a sustained response over that 6-month period. I had mentioned a good safety and tolerability profile, but to save some time, I'm going to move off the safety information because it's very, very similar, if not identical, to what we observed in the Pacific study. I want to tell you a little bit about what our plans are in moving forward. So we've said publicly that we intend to move forward with the global phase 3 program, and said that we intend to meet with the FDA this summer. It is the summer, so we are probably imminently going to be meeting with the FDA to discuss our plans. And I'll just share with you, this is the approach that we're going to talk to them about. The first is a standard approach. This is what most pharmaceutical companies do when they are working in rare or innovative spaces. They do an initial study, and we can go this typical path. This is what's occurred with some of the medications you heard about earlier today. Is first you study Dravet because it's relatively one of the larger groups, and then you can move on to Lennox-Gastaut, and then a third and a fourth and a fifth indication. It's a good approach, but we think there's a better approach, one that's more fair and one that's more equitable, and one that actually gets safety and efficacy information a little bit more broadly. It's an accelerated approach to studying DEs a little bit more broadly. It still starts with an initial study in Dravet, but our second study that would be run in parallel actually would look a lot like the Pacific study that I described, where we would have a much larger study, you can see in the 200 to 300 range, where we'd be studying all patients with developmental and epileptic encephalopathies. We expect to see very similar potential data of what we saw earlier when I was making that comparison of LGS to the D broad group. So you'll need to stay tuned. We'll share our information with the community as we make our plans. And just a little bit of a plug is to think about being involved in research or clinical research for your loved ones. It's the only way we will develop and discover new medications and have other treatment options. Now, our clinical study may or may not be right for you, but being involved in some forms of research, even if it's being involved in registries and filling out surveys, all of that really contributes to our knowledge, and we can get a little bit smarter each and every day. So I think I have a few minutes left, Veronica, for questions. So it's hard to see, so I'm just going to wait for someone just to shout.
Hi, thank you. So for us as parents, caregivers who have to choose medications for our children, can you compare this a little to other 5-HTP receptor agonist medications like Fintepla and Belvic?
So I think the question is, can I compare our study results to other medications? And I think one of the examples you had was Fintepla. Unfortunately, it's impossible to. And the reason for that is we don't run head-to-head studies, especially in early development. So, there may be others that you can speak to, maybe from the folks at Fintepla on direct comparisons, but when you don't run a study at the same time, it's really difficult to make comparisons. Our thinking is that we're working on a medication that focuses on just one receptor, and so I think what we saw in this data set is really good efficacy results that look pretty consistent, not only with other drugs that interact with the 5-HT2C receptor, but also consistent with other medications. But we'll have to wait for bigger and broader studies. Other questions?
I see in phase 3 you're looking at the younger child, ages 2 and up.
Oh, thank you for asking that. The question is about age. So in the Pacific study, we went down to the adolescent population. Subject to our discussions with FDA, we intend to go down to the age of 2 Obviously, we realize the younger patients are those that will have the best chance for potential improvement, not just in seizure outcomes, but frankly, more importantly, in non-seizure outcomes. We expect to go down to the age of 2. The dosing regimen is likely to change and be adjusted, so we need to have that conversation with the FDA. She's coming.
Sorry. Is one of the exclusion, or what excludes you, I missed the first few minutes of the presentation, being on Fintepla to join the phase 3? Would you not be able to be on Fintepla and also be a part of this clinical trial, potentially?
So the question about potential exclusions, such as other medications. In our Pacific study, there were 2 medications that are known to interact with the 5-HT2C receptor, both fenfluramine and lacoserin, so they were excluded. I think it's likely in our planned global phase 3 that the patients would be excluded. Our view is, well, for lacoserin, it's an experimental medication, so we don't think it's wise to have 2 experimental medications be administered at the same time. Mechanistically, they operate the same, we believe. So having a patient on 2 5-HT2C agonists probably, I don't think we'll learn more in that patient population, so it's probably pretty likely we would exclude them.